some of those.
You just have to scream. Yeah.
Try to do is
If it doesn't work, then we'll just repeat your question. But if at least try.
it depends on the.
Oh, it doesn't. It doesn't tell us.
Like when I've been in the all right, so for the other people, yeah, if y'all want to move up, you are more. If you want to just stay there, and then some of y'all might have heard here. We have two people. You all want to do the same thing where you mail rows. That way, get permission every Friday. I don't know if she's going to allow it for the lectures because of the number that are in lecture. It does actually been going to lecture. Barry's now exam block. Yeah. But just email her, and I'm sure she'll try to figure it out in the best way possible. But I know you guys get here every Friday. All right. Does anyone have any questions either on Teams or here before I start with my? Okay.
Hi, Daniel. Will you have office hours before the immuno exam to review the practice exam?
Intrabacterial mobile cell element that can move from plasmid to chromosome or vice versa. This was directly said in his lecture, the transposon. So the nucleoid is just the circular band of DNA that we have in our prokaryotes. Nucleus, you all should know that's going to be in our eukaryotes. And then the plasmid is a piece that is separate from your chromosome that can leave. But the one thing that actually goes between the plasmid and the chromosome, or between the plasmid and the nucleoid, is going to be your transposon. Which type of bacteriophage infection may lead to integration of genetic material into the host genome? So I saw that a lot of people had problems with this one. So the biggest difference between lytic and lysogenic is it's actually going to integrate. Lytic, instead of integrating, it's actually just going to go in and destroy the cell from the inside. So lysogenic, it's going to integrate its own DNA into the host, and then over time, it will then wait, replicate itself, and then kill. So lysogenic is going to lead into killing, but this is the one where we're actually going to integrate into the host cell. So make sure you know that difference. That's going to come up again in block 2 with Dr. Tang when you actually get into virology. Okay, so during which phase of bacterial growth are antibiotics most effective? This should make sense, the log phase, so that's when they're actually growing. Remember, most of our antibiotics are going to be bacteriostatic. Not too many are bactericidal. So if they're bacteriostatic, they're stopping growth. So there's going to be the specific phase that they're interrupting here, which is also known as your growth phase. What would be the consequence of forgetting to add saffron and red to your sample during a gram stain? Also saw people had problems with this one. So remember the steps of your gram stain and what actually is occurring in those steps. So the very first step being adding your crystal violet, which is going to be your initial stain. So that's just going to stain everything purple. Then you add your mordant, which is iodine. What that does is that holds the stain within the actual cell wall. So it's going to hold the blue or the violet into the peptidoglycan. Then you use your decolorizer, so your alcohol. What that does is that washes out that purple stain from anywhere that the mordant actually didn't hold it. So in your gram negatives, which don't have much peptidoglycan, they're not going to actually hold much of that crystal violet. So when you do the alcohol, they get washed out. So at this point right here, if you don't add the saffron in, your gram negatives are just going to be completely clear because they're going to have no stain whatsoever. The whole purpose of the saffronin, which is also known as the counter stain, is to impart some sort of color onto the actual gram negatives. Because at this point right here, before the safranin, they have no color. The only thing that you're going to see is just gram-positives that are going to be purple. All right. All right, this one was also somewhat tricky. Which of the following usually has a mode of transmission passed from one human to another? So when we look at these, rabies is not often going to be passed from a human to another human. This one's zoonotic mainly, so you're only going to see it in that way. Herpes, its highest pass rate is going to be from one human to another. You guys should know that through STDs. And then E. coli. So E. coli was kind of the trick one that I threw in there on you all. E. coli is not always passed from one human to another, but it can be. So this is going to be the fecal oral route. So this is the one where if you work at a restaurant, the entire importance of washing your hands is because if you had E. coli yourself, you don't wash your hands, you touch someone else's food, now it's going from one human, you, to the next that you might be infecting. That's why E. coli falls under this. Zika
Hey, Daniel.
is going to be, this is arthropod vector. So this is going to be the one that he actually explained in his example from mosquitoes that everyone knows from the Olympics in Brazil, I think. Make sure you know those examples. He has them on his slides. He has very specific examples. Make sure you know those examples. I know he spread it out over like 10 slides, and it's like almost nothing on each slide. Try to condense that down and commit all of those to memory. These last 2 lectures that he did,
Daniel?
really, really try to commit that information to memory.
I'm on a call. Hey, Daniel.
Yes. I am able to hear you all. So if anyone has a question, you can ask now.
Can't Zika be transmitted through sexual transmission, right?
So that's not actually, so the thought is that it can be, but the actual vector for it is still the arthropod. So it's still the mosquito, because in order for you to get Zika from the beginning, you have to have gotten it from the mosquito. This one is that one was a little challenging, the Zika one, because there are those outlier cases, which is why I have here usually. So more often than not, it'll be human to human. And Dr. Anderson and I actually had a little debate on that, on this question here, because of Zika and E. coli specifically, which is why the question was changed to usually. So that way we have to go off the most common forms of transmission.
Okay, thank you.
No problem. All right, so this next one, I know this was a tiny, like there's just one slide, and even though it's just one slide, Dr. Anderson did say that you need to know it. He said that to us last year, and very few people committed that slide to memory, and the exact same information from that slide showed up on the test. So make sure you know this information about TIR. So the question, which of the following correctly describes the role of TIR in EPEC? So TIR, what it is it's an actual receptor that the E. coli has within itself. That once the plasmid, once the pili actually bind to the cell it's going to attach to, it sends those TIRs as a receptor for itself. So it's a high affinity receptor for itself that it actually uses to be able to bind to the cell that it's going to try to infect. So make sure you note that. I know it's kind of a confusing diagram. Know the diagram. We'll go over it in my review session on Wednesday and Thursday. But make sure you understand that. It will come up on your test, and it will come up on Block 2 test. Promise you that. EPEC will be on both tests, for sure. And you don't believe me, just go back and listen to the lecture where he pretty much tells you it's on the test. You've never heard him say, commit this to memory until, I don't know, Tuesday? Is that Tuesday?
Daniel, could you repeat the answer for 4, please?
Yes. Give me a second to find 4.
The one about the gram stain.
Yeah, so the answer to that one is the gram negative would not stain at all, because at that point you've used the decolorizer, which removes the crystal violet from the gram negatives. It doesn't remove it from the gram positives because the gram positives have a larger peptidoglycan cell wall, which the stain has been stuck there because of the mordant, which is the iodine.
Thank you.
No problem. All right. So the next question, where would we expect to find the endotoxin LPS and what is its effect? So here in this question stem, you can see endotoxin and LPS. Endotoxin, we should remember from Dr. Anderson's lecture, endotoxin is actually something that is on the cell itself. So it's on the bacteria itself. It doesn't leave the bacteria. So that should automatically get rid of 2 answer choices. So our answer choices as a protein secreted, those 2 answer choices, those both reference exotoxin. So those both reference exotoxins because it's being secreted. So those answer choices as a protein is secreted, those are gone. Not beneficial. So because those 2 are gone, we know it is on the outer leaflet of the cell membrane. And we went through the entire process. We actually even talked about this in the last Q&A session, what LPS does. Its main portion being lipid A works very similar to a cytokine. So it actually works to increase cytokine shock and the cytokine storm. So the one that is involved with cytokines is fever and cytokine release. So that's going to be your answer. Your answer being the outer leaflet of the cell membrane causing fever and the cytokine release. There are other toxins that we see that affect host cell metabolic processes. Those are mainly going to be our exotoxins. When you get to that slide, he has a slide in his lecture about exotoxins, and he has 6 of them. Memorize that slide. Memorize what the toxin is, what the pieces of it, whether it's an A or B, and memorize its function. Please, please do that for your own good. So which of the following regarding biofilm formation is false? So I know he talked about this one very recently as well. So biofilms, in the biofilm, we know that bacteria do communicate via quorum sensing. So that can't be the answer. We also know that they do form an interface between a solid and a liquid. And the biofilms do increase resistance to antibiotics. So the only thing about biofilms here that is false is they are not associated with free swimming cells. They're actually just clumps of cells that then create that film on the outside that protects them from antibiotics. Our next one, which one of the following accurately describes one of the pathogenesis mechanisms of Staph aureus? Oh, that was you. Okay. So to answer this question, you just have to remember Staph aureus and what its toxin is. That's directly from that slide with the 6 different ones. So I believe it has cholera toxin, botulinum toxin, staph, diphtheria toxin. It has all those examples. Make sure you know that. So staph, what it does is releases alpha toxin and degrades the host cell membrane. That's very, very important. I know he asked questions about the one that prevents protein synthesis. You have to know all of these. You have to know all of those toxins and what they do. Really, really make sure you know that slide. All right. And then my last question, which of the following is an evasion? Well, I don't know why I capitalized that high. Which of the following is an evasion tactic employed by gonorrhea? So gonorrhea had 2 specific tactics that it employed to evade the immune system. And both of those are going to be antigenic variation and the IgA protease. Those are the specific examples from the lecture. IgA protease, allowing it to break down the IgA. Remember, that's the immune globulin in the mucosa. And then that antigenic variation, by having those different variations on the outside of its cell, that's what allows it to evade any sort of antibody formation from our immune system. Because remember, our immune system will try to create antibodies to bind to the outside of the microbe. If it keeps changing the genes to the outside, it's changing the receptors on its outside, which then makes it very, very hard to attack and track with our host immune system. Does anyone have any questions regarding mine? Yes.
Could you repeat your explanation for question 3, which details which phase of bacterial growth are antibiotics most effective?
This one? Okay, so the good majority of our antibiotics, we should remember by studying them, they're going to be bacteriostatic. There's very few bactericidal. So like your penicillin, that's going to be a bactericidal, but most of them are going to be bacteriostatic. The definition of bacteriostatic being that it stops bacterial growth, right? So the actual phase where we have bacterial growth is our log phase. So he had a very, very small diagram in his slides where you have the the stationary phase, then you have the log phase, and then a lag phase, and then death. So that log phase is where we're getting a huge increase in bacterial growth. And that right there is where those bacteriostatic antibiotics work. Does that make sense? Okay. Does anyone else have any other questions for?
Yeah. Oh, sorry.
Okay, so on Teams, you can go ahead. Being very nice.
For the EPEC question, how does the lesion form? Like that was the last step.
So the lesion is actually going to be after you have the interaction between TIR and E. coli.
Right.
So that's going to be E. coli actually having its own toxins creating those lesions.
Okay, thank you.
No problem. And you'll get into that a lot more, the different types of E. coli in block. That's an entire lecture in block 2. He just wants you to have an idea of what this EPEC and TIR interaction is now. So that way in block 2, when you go over it, he can go into it a lot deeper.
Okay, thanks.
No problem.
So the question regarding biofilm formations, the free swimming would be planktonic growth, if I'm
Yes.
not mistaken. And can you briefly describe the difference between planktonic and biofilm growth?
So the question in the actual lecture hall, if y'all heard it or not, he's asking on the biofilm question, so the one that I have up at the top, let me put it in the middle. The differences between planktonic growth and the biofilm growth. So biofilm growth is when you actually have, you have that interface between the solid and the liquid. So whether, usually it's gonna occur in the lungs. That's the main place that's gonna occur and that we're gonna see. He has an example in the lecture of that. Make sure you know that bacterial example, the Pseudomonas strain. So what happens there is we get 1 or 2 bacteria that kind of stick to that surface, that separation between solid and liquid. They kind of stick there, and it's through turbulent flow. We'll get into that in physio. So it sticks there. When it sticks there, it's going to release other chemokines to attract the other bacteria to that location, that's the quorum sensing. Once it sends those chemokines, then you get more bacteria growing in that area. As they grow in like a clump, almost kind of like a tumor. As they do that, they're going to be producing that biofilm. And there's no good way to, the best analogy to this is just an awful, awful analogy, but just run with it for a second. Did you ever see March of the Penguins? Awesome, good, I'm gonna talk about it anyways. So in March of the Penguins, all of the emperor penguins just go down and they're in the South Pole and they are in frigid, freezing weather. And the only way that they can protect themselves is by all of them being together in a huddle. So what happens is the ones in the middle are nice and warm and safe. The ones on the outside are getting huge amounts of turbulent wind, freezing, ice cold weather, and they're having a really tough time. So protect the ones from the outside, they go in, the ones from the inside go out. So we keep shifting here. So all it is the actual penguins in that clump are just moving around to protect each other. It is the exact same thing that's happening in a biofilm. So the actual film that's being created by these bacteria is you have a huge clump of bacteria, and instead of the ones on the outside just staying there and creating like a wall, they shift and they constantly move around. And that creates that film that protects them from incoming antibiotics. It also protects them from our own immune response. Because if our immune response or the antibiotics try to go and attack that huge clump of cells, the cells are constantly shifting and moving. So they don't have one target to attack on. With the other type of growth, that's going to be more seen in a liquid vector, so like in your blood, so sepsis, or in a purely solid medium, so your skin. So when you have a normal bacterial infection on your skin, you have a normal Staph aureus infection. You'll have one that's going through and it's going into that log phase. And in order to get into that log phase, it needs resources. So it's stealing the glucose from your cells. It starts growing, it starts dividing, and creates more and more and more. Even though it's creating more and more and more, they're not creating that little penguin huddle to protect each other. So they're just there. So now with antibiotics or our immune system, they can attack and attack from the outside in. That makes sense? So, no, yeah, you're gonna sit there and you're gonna think March of the Penguins, and that's gonna be the best way to remember that. Anyone else have any other questions?
Hey, Daniel,
Yes.
could you review the evasion tactics for gonorrhea again?
Yeah, so the main 2 that he discussed in lecture, and these are really the only 2. There is a rare chance that it'll be a capsule, but that's extremely rare in gonorrhea. So the main 2 that he spoke of were antigenic variation and the protease, the IgA protease. So the IgA protease, it's just cleaving proteins, and what it does is it breaks down our human immune globulin IgA. So it destroys our antibodies that we find in our mucosa. So because it has this protease, that means IgA cannot attach to gonorrhea and can't enact its own function onto gonorrhea. So that's one way it protects itself. It stops IgA from actually doing its own function. The other, the antigenic variation, this is essentially I'm just going to use another odd reference here. So let's say gonorrhea is just wearing, I don't know, a black hat. That's what we're going to go with. So its variation on its outside is a black hat. If we tell our immune system, which is going to be similar to our police force, hey, we're looking for a guy with a black hat. Gonorrhea has a bag of 30,000 different hats that it's going to take off and put on. So now your immune system is looking for someone with a black hat. Gonorrhea is going to change its outside surface, put on a red hat. Then your immune system says, okay, now we're looking for someone with a red hat. It's going to do the exact same thing. It's going to take that red hat off, put a blue one on. So it's going to keep changing its outer appearance so that way it can avoid our immune system. Does that make sense?
Yeah, that makes sense. Thank you, Matt.
Anyone else have any other questions? All right. So, yes. So, Phoebe, are you there? If you are, you have the con, and it is your questions now.
Awesome. Can everybody in the auditorium hear me okay?
Yeah, you're good, Phoebe.
Okay, thanks. All right. So for the first question, a researcher is measuring the amount of product yielded from an enzymatic reaction per unit time. He adds 20 micromoles of substrate to a constant amount of the enzyme and notes the reaction velocity to be 40 moles per second. When he increases the substrate to 100 micromoles, the velocity remains the same. So which of the following is true about this reaction? So most of you got this correct. It is a zero order reaction. Why? Because when you add more substrate, the reaction is not going to change in the velocity, and that means that the substrate concentration or the rate of the reaction is independent of the substrate concentration. So that right there is indicative of a zero order reaction. Zero order reactions also do not form a hyperbolic curve. Remember, those are linear, and they're independent of the concentration of substrate, so that's why the last answer is incorrect. So the next question, when the affinity of the of the enzyme for the substrate decreases, but the maximum velocity of the reaction remains the same following the addition of an inhibitor, it is known as a competitive inhibitor. So remember that when you add a competitive inhibitor, it's going to decrease the affinity for the substrate, but the Vmax is going to be the same. So that's just, going to be a competitive inhibitor. It's not going to be an irreversible inhibitor or an allosteric inhibitor because it's binding to the active site. And then the non-competitive inhibitor would be the answer if we were talking about the maximum velocity decreasing with the affinity staying the same. So remember, affinity is associated with the Km value. So the question after that, when the concentration of substrate is below the concentration at Vmax, the reaction rate is proportional to the concentration of substrate. Knowing this, which of the following is true? So the key phrase here is that the reaction rate is proportional to the concentration of substrate. Right away, you know that this is going to be talking about a first order reaction. So Roman numeral number 1 is going to be true. That makes Roman numeral number 2 false. And remember that with a Michaelis-Menten plot, you're going to be plotting the initial velocity for each of these reactions at a different substrate concentration. So that makes Roman numeral number 3 correct. Roman numeral number 4, that states the velocity is constant, that's going to be incorrect because that would be true if we were talking about a zero order reaction. So that makes number 1 and number 3 correct, and it looks like 37% of you got that correct. Okay. So the next question is, all the following are assumptions made by the Michaelis-Menten equation except The maximum velocity occurs when the enzyme is fully saturated. We know that to be true. You get Vmax when all the enzyme active sites are full of enzyme and you cannot increase the velocity anymore. The next one is the substrate concentration is significantly larger than the enzyme concentration. This is also true because remember, in order to measure this kinetic reaction, you need to have it such that there's less enzyme than there is substrate. And then the last option was the concentration of enzyme substrate complex is constant. Remember, this is the steady state assumption, and we use the steady state assumption in order to create the Michaelis-Menten equation. So the first option, which is the false statement, the maximum velocity occurs when equilibrium is established. That is false because remember, we're not at equilibrium at Vmax. It would be if we were plotting product concentration on the y-axis over time. That's when we would reach equilibrium, when you get to that point on the graph where the product doesn't increase or decrease. So don't mistake the Michaelis-Menten plot with the product over time plot. Okay, so for the next question, we have for a cooperative enzyme which such as ATCase, which of the following must be true? So this is just trying to get you to understand that at the R state, which is the relaxed state, you're going to have a high affinity for substrate. So you would have at a high substrate concentration, most of the active site is going to be filled with the substrate because of the high concentration. And like we talked with hemoglobin, when you have more substrate bound to the enzyme, you're going to be more receptive for accepting the substrate. So you're going to have more of the relaxed state conformation for the enzyme. On the contrary, at a low substrate concentration, you're going to have less substrate bound to the enzyme. And because less substrate is bound, you're going to have a lower affinity for the substrate, which means that the enzyme is going to be in the tense state. So it looks like most of you got that correct. Just, make sure you get your T state and your R state figured out. Tense state is going to be lower affinity. R state is going to be higher affinity. So for the next question, a patient is prescribed warfarin following his diagnosis of atrial fibrillation for prevention of blood clots. Which of the following will be directly affected after this patient starts taking this medication? So remember that warfarin is going to be a vitamin K antagonist. And you have to know that the vitamin K is going to be a cofactor for the reaction that makes gamma carboxyglutamate. So if you were to not have gamma carboxyglutamate, your prothrombin will be unable to bind to the platelet membrane, and therefore it won't be able to associate with factors 5 and 10, and so therefore your prothrombin will not be able to be converted to thrombin. So that is why the prothrombin will be unable to bind to the surface of the platelet is the correct answer. The first two options deal with fibrin. So while yes, downstream the fibrin will be affected because you don't have thrombin to activate the conversion of fibrinogen to fibrin, I was asking for the direct effect, and the direct effect would be the prothrombin binding to the platelet for activation. And then the activation of plasmin will be inhibited is false because plasmin is like way later down the line. Okay, so the next question, which of the following is the correct order of the 4 phases of hemostasis? So this is essentially the first slide of the last lecture. And so the order is initially you have your injury, and then right after the injury, you're going to have your vasoconstriction to minimize blood loss, followed by the platelet binding. So that's the second step. After that, you'll have the zymogen activation. And the zymogen activation is all of that blood clotting cascade that we went into detail with today. And then the last step would be the dissolution of the clot, which is just the breakdown of the clot after the injury is healed. So make sure you know the order of that. Okay, and then the next question is, what occurs when thrombin catalyzes the formation of fibrinogen to fibrin? So remember that this is that one slide where you have the 3 polypeptide chains, your alpha, beta, gamma. So remember that thrombin is only going to cleave the amino terminus of the alpha subunit and the beta subunit. So the correct answer is the amino terminus of the alpha subunit must be cleaved to remove the negative charged amino acid residues. If I had put the amino acid terminus of the beta subunit, that would also be correct. The alpha and beta subunits will not be cleaved off entirely because remember, if you cleave them off entirely, that would just leave you with just the gamma chain by itself. It's only the amino terminus that is cleaved off. The same thing for the next question. If you were to cleave one of the polypeptides to turn it into a dipeptide, that's also not true. And the fibrinogen must be cleaved into its soluble form is false because remember that fibrinogen is the soluble form. It's the fibrin that is the insoluble form. So yeah, that's why A is correct. Okay, and the next question is, which of the following is the correct sequence for the final common pathway of the blood clotting cascade? So essentially, this is just telling you the order in which all of this occurs and what factors activate the next factor. So remember that it's going to be factor 10 and factor 5 that are on the platelet membrane that prothrombin will associate with to activate thrombin. So after thrombin is cleaved from its propeptide sequence, it's going to float into the blood and ultimately activate the conversion of fibrinogen to fibrin. So that's why the first option is correct, because that's the correct order. And then remember that it is factors 10 and factor 5, not 10 and 13, which was the case for 2 of the wrong answers here. Okay, the question after that is, all the following are required for thrombin activation except, so this is just your prothrombinase complex, so it's those 4 things that you have to memorize. So factor 9 is the correct answer because it is actually factor 10 that's part of the complex, not factor 9. All of the rest of them are true, so you'll have calcium, phospholipids, and factor 5 that are part of this complex. All right, and the next question is, between 2 amino acid residues, does transglutaminase form crosslinks for the formation of fibrin? So this is just another memorization for the slide that is talking about the formation of the crosslinks. You have to know that it's the glutamine of one fibrin molecule interacting with the lysine of another fibrin molecule to form those crosslinks between the fibrins and kind of stabilize the whole structure. So yeah, just memorization. And the last question, I gave you a graph of the Michaelis-Menten plot. So the following graph is demonstrating the addition of 2 different types of inhibitors to a normal enzyme that follows Michaelis-Menten kinetics. Which of the following is true about the blue line? So from the blue line, you'll see that it doesn't ever reach Vmax. So right away, if it doesn't reach Vmax, you know that it's going to be a non-competitive inhibitor. So that's why the last option is the correct option. So the first option that states the blue line is showing the addition of a competitive inhibitor, that's incorrect because the competitive inhibitor would look like the red line, where it takes a little bit longer to reach Vmax, but it is still able to reach it. The second option, the blue line has a different Km value than that of the red line, is incorrect as well because remember, for non-competitive inhibitors, you will be unable to reach Vmax, but you still have the same Km as that of the uninhibited enzyme. Whereas for the red line, which is showing a competitive inhibitor, you're going to have the same Vmax, but you're going to have a different Km. So between the blue and the red line, they're not going to have the same Km value. The next option is the blue line is dependent on the concentration of the substrate in order to reach Vmax. This is also false because remember that with a non-competitive inhibitor, you cannot oversaturate the solution with the substance because you cannot oversaturate it and cause there to be the Vmax to be reached because you can't what's the word I'm thinking of? You can't oversaturate it. You can't knock the inhibitor off by just adding more substrate like you can with the competitive inhibitor. So that's the end of mine. Does anyone have any questions about any of those?
Phoebe?
Yeah.
Can you explain the difference between a non-competitive inhibitor and an allosteric inhibitor?
So they're like they have like minor differences between the two. So kind of categorize them differently. So with a non-competitive inhibitor, they both have the binding to the allosteric site. But when she talks about like allosteric inhibitors, she's more so talking about allosteric regulators that are typically found in your body that, like the example she gave you that cooperative enzyme that has multiple subunits, that has regulatory binding sites for both allosteric effector or activators and allosteric inhibitors. So this is more of like a natural thing that occurs in your body, and also is most likely found with cooperative enzymes that have multiple subunits. So it's not going to have the hyperbolic curve that you would see with a non-competitive inhibitor. You would see a sigmoidal curve for the allosteric regulators and allosteric inhibitors. Additionally, I think we see non-competitive inhibitors more so as like when we go to, I think pharmacology, you'll see it with like the addition of certain drugs. So it's like not something that's natural versus like allosteric regulators with like your CTP and all of that as part of like your body's regulation system for different enzymes. So definitely categorize them differently. She definitely mentioned that you should know that allosteric regulators do not follow Michaelis-Menten kinetics. So they're also different in that regard.
Thank you.
You're welcome. Any other questions?
There's one in the lecture hall.
Okay.
The question is, can you explain why they have different KMs? Or
Okay.
why they don't?
Yeah, so remember that the Km is equal to half of the Vmax. So if the Vmax of the competitive inhibitor is going to be the same Vmax that you would have with an uninhibited enzyme, your half Vmax is going to be the same, and therefore your Km value is also going to be the same. So if you have a non-competitive inhibitor, now you're not going to have the same Vmax as you would with an uninhibited enzyme. So your half Vmax is going to be lower than that. And as a result, where the non-competitive inhibitor's line intercepts with the half Vmax, it's now going to be lower, and therefore your Km is also going to be a higher number. But you can also rationalize that, because with a non-competitive inhibitor, automatically that, with a competitive inhibitor, automatically that your Km, which is standing for the affinity of the enzyme, is going to be impacted, whereas the affinity of the enzyme is not going to be impacted with a non-competitive inhibitor. Does that answer the person's question?
Yes, I think so.
Okay. There is a question in the chat, so I'm going to answer that. It reads, for your first question, I was confused because I was thinking if a first order is at Vmax substrate concentration at the 20 micromoles already, then the velocity still wouldn't change, right? So yeah, if you were at Vmax, for the first question, I tried to say that you were below Vmax by saying you're at below half Vmax. I think I wrote it in a way that would tell you that you are below the Vmax. So you would be doing a first order reaction, because I tried to specify that you were like under the Vmax. So hopefully that makes sense to you, Veer. Jaylene, which one was question 19?
It's the one regarding the pathway of the blood clotting cascade.
Okay, is that the one with the arrows?
This one right here? Bingo. I think I got it.
It's that one, Jaylene?
That one, yeah. Yes, it's that one.
Okay, so this is just looking at that one slide that she repeats over and over again of which factors activate the next factors, and it's just going in order of like what's activated first. So you know that factors 10A, which is the active form of 10, and factor 5A, the active form of 5, they are associated with the platelet, so they're bound to the platelet membrane. And that's why when prothrombin binds to the platelet membrane, it can get the prothrombin complex, it can get the prothrombin close enough to these two factors for them to both kind of work together to cleave it into the active form of thrombin. So it's the factors 10 and factors 5 that activate thrombin, and then you know that thrombin is then going to catalyze the it cleaves the fibrinogen into fibrin. So it's just like saying like what the order of these events are. Does that make sense, Jaylene?
It does, thank you.
You're welcome. Any other questions?
We have one in lecture.
Okay.
Why do we need to have more substrate concentration than enzyme concentration for the assumptions of, what'd you say, Nicholas-Menten? For this question? Or, wait. We accept, okay. Yeah,
Okay.
so, yeah, okay.
Yeah, so the reason for that is because in your body you have way more substrate than you have enzymes, so it's kind of trying to mimic your physiological conditions. Additionally, if you have like close amounts of enzyme and substrate, it's not going to follow the same kind of like pathway. So it's really just to mimic your physiological conditions for the most part, so that it could follow these same kinetic rules. Does that make sense?
Yep, we got it. Thanks.
Cool. Any other questions?
Maybe check your phone.
I have a question, sorry. Can you go over 22, the one with the graph again? And can you explain how the blue line doesn't have a different KM than the red?
Right. Okay. So you see how the blue line kind of has a similar slope to that of, I think the drawing's a little bad, but honestly, it's supposed to have a very similar slope to that of the normal enzyme. So when you divide the Vmax by a half, it's going to still be proportional to the substrate concentration. So your Km, which is going to be on your x-axis, if you were, I wish I could draw, but like if you were to draw a line from your half Vmax, and if you were to, if you draw a line to the new Vmax that the blue line makes, you'll see that if you take half of that, it's going to be the same Km value as the uninhibited enzyme, because it has like a similar slope. So it's moving kind of at the same rate, versus with the red line, if you take half of that Vmax, you'll see that it doesn't have a similar slope. It's not changing as quickly because it's inhibited. So the half Vmax will be kind of shifted over a little bit. I have it in my PowerPoint slides, kind of that whole thing kind of drawn out if you want to look at it. I wish I could show you, but I can't. So I hope that was a good explanation.
Okay, yeah, I'll take a look at your PowerPoint slides, and then I'll like try to make sense of it, and then I'll email you if anything.
Yeah, awesome. Sorry about that.
Can you guys see what we're drawing on the screen if we're sharing the screen with you, or no?
I can't see anything.
No? Okay.
Thanks for trying, though.
All right. Any other questions for Phoebe in lecture? No. Okay. Any more questions? Oh, 13. We need to go over 13 again. Which one is that? Am I even on the mic? Yeah, okay. Which one is 13? The third, oh yeah, okay. Right. So, 1, 2, 3, got it.
1 and 3.
Phoebe.
So what's the question here? Sorry.
I guess just explain this one.
Oh, okay. So the question is, when the concentration of substrate is below the concentration of Vmax, the reaction rate is proportional to the concentration of substrate. Knowing this, which of the following must be true? So if you are below Vmax, we saw that when you're below Vmax, your reaction rate is going to follow a first order rate. So you're going to have a increase of the rate proportional to the amount of substrate increase. So I hinted that it was going to be below the Vmax by saying that it is below the Vmax. So you should be able to understand that because it's proportional to the concentration of the substrate, right away you should know that we're talking about a first order reaction. So that's why number 1 is correct, and why numbers 2 and 4 are incorrect, because your velocity is constant only in a 0 order reaction. The initial velocity of the reaction is being plotted at different substrate concentrations. So this is literally just explaining the plot of the Michaelis-Menten graph. So when you plot this graph, as I kind of indicated, when you're plotting to find the Vmax, you're going to be plotting the initial velocities for each of these substrate concentrations, because remember that the initial velocity is going to be the fastest velocity of the reaction. So we're only plotting the initial velocities for each of these substrate concentrations on a Michaelis-Menten graph. So that's also true because I'm referring to a Michaelis-Menten graph.
We're good. Any more questions in here? Okay. I guess that's all for you, Phoebe.
All right, cool.
Okay, then I'm just going to take over. Okay. So my first question, how many base pairs is a read? So that's going to be 150. He kept saying 120 in class, but then the chart on the slide said 150. So if that were to show up on your exam, it wouldn't be like 120, 130, 140, 150. Like it would be kind of like how I did it, with none of them being that close. So you have to know that he likes to test you guys on certain numbers. So then the next question, true or false, when conducting massively parallel sequencing, the DNA fragments must be overlapping. So that's true because we can't sequence our entire chromosomes all at once. That's why we have the reads. That's why we have to fragment them. But we can't just fragment them and not overlap them, because then we can't like put them back together and figure out what the actual sequence was. So they need to be like overlapping so we could kind of put together like puzzle pieces and figure out like which ones, what does this mean? Okay, if everybody could still see, then that's good, and I'll ignore it. Okay. Traumatized with technical difficulties. Okay. So which of the following sequencing methods is not second generation sequencing? So that's going to be the dideoxy or Sanger method. So we're only doing one template at a time when we do this DNA sequencing method. So that's why it's good to like check to make sure, like double check something and make sure, but not to sequence like a lot of things at once because it's more time consuming. So that's why we have these massively parallel sequencing techniques. So like Illumina and NextGen, you can do multiple templates at the time, and that's why we call them massively parallel. Okay, which of the following statements is incorrect regarding Illumina sequencing? So the answer was the fluor interferes with Watson-Crick base pairing. Looks like a lot of you guys got that right. So it does not interfere with Watson-Crick base pairing. If that was the case, then this method would be pretty stupid because then we can't actually sequence. So we can remove the fluor because that's how we make sure we're not getting every single nucleotide fluorescing whenever we add a new nucleotide. And the 3' OH block can be removed. That's what makes this Illumina sequencing different from the dideoxy. So we can remove that block and then we're able to add on a new nucleotide after that. So that is true. And then we must stop after every nucleotide. That is also true because we're stopping to take that picture so that we can compare and then we can like make the sequence from there. So all of those are true except for the first one. So what type of linkage is between the primers and the chip in next-gen sequencing? So that's going to be the covalent linkage, and that's why we call it PCR in place. So like they're not moving around on the chip, like each sequence is for that specific like transcript that we're doing at that spot. So you know that it's always going to be that sequence for that specific DNA fragment that you're trying to sequence. So PCR in place, they are covalently linked to that chip. They're not moving around. True or false, the segment of DNA incorporated into the bacterial DNA with an origin of replication in the recombinant DNA sequencing method cannot be separated out from the bacterial DNA. That's false. Looks like most of you got that. So we said the steps were like amplify and isolate. So we're using that bacterial vector with its origin of replication to amplify that DNA sequence, and then we have to isolate it after that. So we can isolate the DNA from the bacteria. Glioma tumor cells with silenced MGMT genes have a blank at the MGMT regulatory region. So that's going to be an HDAC. If you remember what the function of HDAC is, please remember what the function of HDAC is. That like is probably going to be on your test. Like you just have to know that. So we know that it's going to condense the chromatin. So if we condense the chromatin, those MGMT genes are silenced. We know that the glioma tumor cells do that. It's like favorable for them. So yeah, only 2 people put Electric Feels. It's a great song. Okay. So true or false, the trans configuration of thymine dimers disrupts the helix much more than the cis configuration. So that is true. The trans configuration disrupts it more, and that's why it gets like repaired more efficiently, because our DNA repair mechanisms can recognize that disruption way more than the cis configuration. So it gets repaired more efficiently than the cis does. So glioma tumors are more responsive to mutagenic therapy because, so if you understood this question, then hopefully you understand this question. So they no longer have those DNA repair mechanisms, so it might be somewhat favorable for them to silence those genes, but then we can take advantage of that and we can induce like this mutagenic therapy. And if they no longer have those DNA repair mechanisms, then they can't repair that DNA and they are effed. So, okay. Guys, let's keep our mics muted, please. Okay. The XPC protein is a part of which repair mechanism? So you kind of just have to know, like he gave you a list of certain proteins that he wants you to remember. So you just have to be able to associate those with their repair mechanisms. And this is like the first one that we go over that does disrupt that phosphodiester bond. So just make a note of that. And just make a note of this XPC. He didn't want you to know all of the other XPs, but there are more XPs obviously, and they all correspond to this xeroderma pigmentosum disease. So if this XPC is defective and it doesn't recognize the distortion, then you're going to get this disease, xeroderma pigmentosum. And he said on in his lecture on the test, he probably won't ask you about the other XPCs, but if it is an XP something, then you know that it's related to this disease. Okay. So true or false, uracil DNA glycosylase cuts the phosphodiester bond. That's false. What bond does it cut? Somebody tell me. Yeah, glycosidic bond. So I thought somebody typed something. Okay. So you have to know that difference. That's why he wants you to know like which bonds are which. He wants you to know the numbering system of the sugar. Make sure you guys know that. Okay. I'm going to also upload for you guys like a document of like stuff to go over, like helpful topics that you should look over and like kind of what material you should be looking over. So I'll probably send that out tonight. So definitely look at that. Okay, questions. Could you go over 29 again, please? It talks about the glioma tumor cells being silenced. Oh yeah, the electric fields. Okay. So these MGMT genes are repair, like it's a repair mechanism. So if we don't have this, if it's silenced, whatever mutation that caused that cell to become a cancer cell, that mutation is not going to get repaired. So the cell's like, yes, I'm going to become a cancer cell. I'm silencing these genes. This is more fun for me. But oh, so that's why we have an HDAC at those MDMT genes to keep them silenced. But we can exploit that and induce mutagenic therapy, and then they can't repair their DNA anymore because we silence this MGMT gene. Does that answer your question? You're welcome. Okay, does anyone in lecture have a question for me? Dang, am I that good of a teacher? No, I'm just kidding. Yeah. So like on my slides, I have like, whenever he gives you an example like that in class, I've written them in red and I've written like exam cue. And then I tried to write the question and the answer that he gives you in class. So definitely like when you're done going over all the material, like go back and like scan for those and like do the fill in the blanks. He will pull questions from there. He's done it for us. He also, I guess, gave you guys some practice questions. So I was kind of scrolling through the discussion board to see if he was answering you guys because I kept saying it all the time that he does. So I was looking and I saw that he did answer you guys and I saw that he put like some questions. So scroll through there and find the questions that he gave you and then maybe post like what you think the answers are in the discussion board so that he can answer you. But I would do that too. In the discussion board, I saw he linked some questions for you guys. So yeah, okay. Regarding question 23, base pairs range, one of the slides says each individual sequence read is relatively short, approximately 200 nucleotides. Is that going to be the answer or do we ignore that? No, he kept saying 120 in class. And then like once he got to whatever slide it was that it was in like that chart, he was like, oh, I was a little concerned. Than the text. You probably wouldn't even put 200.
So when you were talking about the ribose numbering system, they just memorize it just like all the carbons are saturated with hydrogens except for like the prime.
Yeah, 4' gets left out. We don't really care about her, but we care about C1, C2, C3, and C5.
Wait, which one did you say leave out?
C4.
Yeah, they're all the same except 5.
Well, so you have to know where the glycosidic bond is taking place. You have to know which carbon, if it has an H versus an OH, is determining if it's a ribose or deoxyribose. Then we know that we always have an OH at the 3' carbon, except for if we're doing like dideoxy, it's removed, or Illumina, it's blocked, and then 5' is the
Okay, so
phosphodiesterase.
this ties in with like the very first lecture.
Yeah.
Okay, cool.
And he will bring that back.
All right. Yeah, I didn't know if it was like a separate.
No, from the first lecture. Yeah. Any other questions, guys? Okay. All right. Well, then
Hey,
that's, yeah.
sorry. I had a question. If you go back to the one about glioma tumors are more responsive to mutagenic therapy. So I remember Dr. Blank talking about the lack of DNA repair mechanisms, but is the idea that you're just trying to like induce mutation or like create mutations that'll induce apoptosis? Is that like what the goal is?
Yeah, so we're going to induce mutations, and since those cancer cells don't have those DNA repair mechanisms, it like can't function anymore with those mutations, so then it just dies.
Okay, so the specific mutations are meant to like inhibit normal cell function altogether, and then the cell just dies.
Yeah.
Got it. Okay, thank you.
You're welcome. Anyone else? Any questions like regarding the exam block? Any general questions? How did you guys feel anatomy went? Good, bad, so-so? Wasn't
Better
too
than I
much.
thought.
Nice. Okay, good. That's good. How do you guys feel for the upcoming week? Like ass. Yeah. Okay. It's gonna be okay, guys. You're almost there. Well, personally, I thought that genetics and biochem were like the easier ones. So do with that how you please. Okay, well, people thought genetics was hard because they overstudied and they overthought the questions. So I've tried to like prevent you guys from doing that. Elizabeth, you're scared of which one? Genetics? Don't be scared of genetics. Dr. Blank is actually super helpful because he gives you those fill in the blanks. He gives you like those example questions. The questions he posted, yeah, there's only like a few. Oh, I cannot see that from here, but yeah, he just like linked some questions in the discussion board I had saw. Okay. Oh yeah, I'm waiting for him to review my practice exam before I publish it for you guys. So hopefully it will. And the immuno one will be published in like 2 hours. So, you know, he likes to go over it. We like to send it to our professors to make sure that they're good practice exams. So. Oh, you're welcome, Elizabeth. We got a compliment. Okay. I mean, if you guys want me to, I could just tell you what I was going to say for what to do before the exam. So, you know how he makes those lists on his slides? Okay, make sure you know everything on those lists. He likes those lists. Okay, I already told you about his practice questions. Okay, do his fill in the blanks. Go back and do these Friday Q&A questions. So like, if you didn't know them, whatever week that you did them, hopefully you know them now. Do my practice exam. Like I said, he's gonna review that and then I'm gonna publish it as soon as he's done. Look at my documents that say important things to know. Those are like my notes that I took from my TA last year, and he would just like highlight stuff on this document and I would like take things out that I didn't think were important or put things in that I did think were important. So look at that. Okay, go through my lecture notes and find those example exam questions that I've written in red. Come to my office hours before the exam, and then email me with any of your questions. And then I'll send this out so you can see like the important topics from like each lecture to look at. But okay, if there's no more questions, then go study. My office hours, I'll send out an announcement. What did I say?
Ivy,
Yes.
what happened to your cat?
Oh, she's doing better. I mean, this is the first time I've left her home alone, so hopefully she doesn't get her cone off and start licking her incision, but she's doing better. She started to walk now.
Oh, that's good.
Yeah, thank you for asking. She's actually doing a lot better. Oh, I had taken a picture of my... No, because I didn't include my office hours on that. Because I wasn't... Oh, aha, I found a picture. Okay, so my office hours are going to be the 29th from 6 to 8, and then the 30th, which is the morning of your exam, I'll do like a last minute questions from 9 to 11, and then I'm obviously always available through email. And then we have our last session at 5 p.m. after your exam on Monday. And then at 9 a.m. the next day, September 29th, we're doing our last session. Okay. I have a question. Professor Blanc posted a document with what he would suggest us to put on flashcards. Would you say we should literally put those things as he said? Or do we have to elaborate on the points he made on the doc? Yeah, I would elaborate. Yeah, just it's better to be like a little bit over prepared rather than under prepared. So like whatever he said in lecture, like you just know that. So, okay, you're welcome. Yeah, nobody else. I'll stick around for a few minutes in case you guys think of something. Oh, look at my kitty. He's so cute. I don't know. He just like, yeah, he just like posted like some responses, I guess, and they were like linked there. I was just scrolling. Yeah, he did send out an email. I didn't know if they were the same questions because I didn't open them. Oh, okay. I just saw that he put that there, so I was like, I'll tell them to look. Okay. Yeah. He does love the discussion board, but I know like it's hard to like get instructor interaction through the situation, so that's why I keep preaching it because I know he actually checks it. He'll comment. You have to like scroll down. Yeah. You're gonna have to kind of get used to discussions because in ethics, that's basically the entire course in summer. No, it's not an elective, it's required, and it's Dr. Barber's course in the summer. Yeah, ethics. It's not a bad class. Yeah. Yeah, like lecture 8 is what you're asking? Yeah, so that's lecture 8, and then lecture 9, I'm like finishing that up. I'll probably post that like either today or tomorrow. And then depending on what Dr. Blank says about my practice exam will be tomorrow. And then we're going over the last lecture, lecture 9, instability of the human genome, and the second highlight from the articles that he posts. There's 2 you have to know for the exam. So we'll go over lecture 9 and the second article on Monday if you come to the 5 p.m. If you go to the 9 a.m., it's the next day. Kind of cold in here. Yeah, if you don't have any questions and you don't want to stick around, then don't feel bad leaving. But if you want to stick around. What types of questions does he ask for the articles? I'm not really sure like how to answer that because like whatever he has highlighted can be a question, you know? Like he can ask like what like should we just memorize the information that's highlighted or is it comprehensive? No, like so if you look at the document I posted for the first article, the telomeres article, I just kind of like organize the information a little bit better for like what was highlighted. So like for example, I wrote like telomere dysfunction, shortened telomeres has been found in some disorders. And then I wrote like what the typical manifestations of those disorders are. Then I wrote like what telomeres are, like discarin is an essential component of telomerase. Then I wrote like what genes, so he could ask you about like the TERT gene. So that provides instructions for making a component of the enzyme telomerase. So like maybe he could ask you like what gene is mutated in telomerase? And then it would be like a bunch of genes that don't relate, and then it would be like TERT. So he could ask something like that. He could ask, so like there was also something that said there's an average of 92 telomeres in an interphase cell. He could say like, on average, how many telomeres are found? Whatever, and then 92 would be the answer. So just try to know like the main idea of like those highlights. And he likes numbers. You're welcome. Yeah, I said like one could be like what gene is like mutated. Like if you have a telomere disorder, like what gene could be mutated, and then it would be TERT with a bunch of other genes that aren't related to telomeres. Or it could be like on average in a human interphase cell, how many telomeres are present, and then it could be like 92. You're welcome. Yeah, so I would definitely go back and look at like also the questions that I have for my TA sessions. Look at those too, because I counted them after I made your practice exam. I'm pretty sure in total it's like 130 questions that I've made for you guys. There's only like so many topics. You know what I mean? So if you do like all those questions and you understand the concept in its entirety, not just necessarily the question, then you should do pretty well. Yeah.
Yeah. How long do grades usually take? A day? Like next day, 2 days?
That's a hard question to answer because like during fall, it would take like a few days to get them back. Same with during spring, but then like in summer, we would instantly get our grades. So I don't really know what they're gonna do. You might get them all like once the block is over, or you might get anatomy like in the middle of next week, like randomly. So like just be prepared for that.
Okay, thank you.
You're welcome. Yeah. So massively parallel sequencing is like when you're doing more than one transcript at once. So like everything that does more than one transcript at once is like massively parallel. So it starts with second gen, which is like Illumina sequencing, and then also next gen, which is the one that has like the bridge amplification, like where it's on the chip and then it like makes a little bridge and then it gets amplified, whatever. That's also massively parallel. He kind of didn't really like say that. That's what I took from my TA, and that's how I went into it, and that's how we all interpreted it. Yeah, because massively parallel is just like you're doing more than one transcript at once. So you're doing them in parallel, and it's massively because it's so many at once. So like the only one that's like not doing that is the first one, which is the dideoxy Sanger, because you're just doing one at the time. Yeah. Where are the grades posted? If you go to, yeah, they're not posted right now, but where's my mouse? Oh, if you go like on the side of your thing for each class, you can click grades and it will show up there. You can see like the high, the low, and the average. You're welcome. Yeah, yeah, you're stopping after each nucleotide, but you're doing all the transcripts that you want at once. So yeah, you're stopping every time, but you're done like when you're done. You're not just done with one transcript and now you have to do it all over again for another transcript, which is why the dideoxy is good for like checking something. So it's like, oh, I think you have a mutation here. You might have this genetic disease. Let's double check. So now we know specifically what sequence we want to look at, and it's more accurate as well. So then you're like, okay, this is a specific sequence I want to look at. I'm only doing one at the time, and I'm using this because it's more accurate to double check. And then, oh, I did have that mutation. Okay, you do have this genetic disease. Yeah, they're still pretty precise, but yeah, they're quicker. Yeah, you're welcome. And then also remember that the back clones, those can have larger fragments. I don't know specifically why, but just know that it's like larger fragments. So like the reads that we've been talking about are like 120 or 150, whatever number you want to go with. But the back clones can have larger fragments. Just know that. Yeah, he said it in class. Sometimes if you've noticed, we know that a lot of things are true and we don't necessarily know exactly why that's the case. So maybe you could ask him and maybe he knows why and he didn't like get into it because it's like kind of complicated, but just know that.
Hi, can I ask a quick question? I'm sorry.
Of course.
I know you asked like what was the read, right? You said it was 150.
Mm-hmm.
How about the other one he spoke about? What is an exome?
The exome is all of your exons in your entire genome sequenced, excluding every other sequence. It's only the exons.
Okay, thank you.
You're welcome. No, he just put it in this chart.
Hi, I'm sorry, I have another question.
Yeah.
Can you help me understand the contigs in like one chromosomes?
The contigs.
Yes.
So the contigs, hold on, let me pull it up so that I can go through it with you. Good thing I brought my iPad. Okay. So contigs, okay. So we can't sequence our chromosomes like in their entirety. We have to fragment them. So we fragment them. And with these bacterial clones, we can have larger fragments with like more overlap than reads. So we take all those overlaps and then we piece them together. So if you go on to slide 26, we can see that we have all these different fragments of 1, 2, 3, 4, all the way until 21. So once we piece those fragments together and we line them up and we put the puzzle back together, like, okay, we know that that's the order that they go in, we refer to that as the contig. So contigs represent large portions of chromosomes. Where the entire sequencing project is complete, the entire genome is represented by one contig per chromosome. So it's just like matching up all those fragments that we sequenced.
Okay, thank you.
You're welcome. Oh, really? Well, I was cracking up because in my undergrad genetics course, he gave us a question about the MGMT, and he put not the band that sings that song, whatever. Yeah. And I didn't know the answer. And I couldn't remember the name of the band. And it was like a fill in the blank. So I was like, well, I'm effed. So that's why I included that for you guys, because I thought it was kind of funny. I'll never forget that, because I was like, he literally gave us the answer, and I just can't remember the name of this band.
I haven't been told. I don't think they're going to. We had that on a few where it had the name.
Hi, I have a question. I have another question. I'm sorry.
Okay.
Do you mind go over with me the RNA transcription versus the IVS gene, I think, in human?
You mean the difference between IVS and introns?
It's IVS, intron, and in RNA transcription.
Okay. Well, if you're asking about the IVS, okay. So we know that ribosomes are usually two pieces. They have two subunits, a large subunit and a small subunit. So we have our 18S and our 28S genes. Those genes are making the two different subunits. So when we transcribe the human ribosomal RNA, we're not going to splice the 18S and the 28S together when we do transcription. So you know how we normally cut out the introns and then splice together the exons. So we're not going to splice them together because they need to be processed separately because they're making two separate proteins, the large subunit and the small subunit of the ribosome. So since we're not splicing them back together, that DNA sequence in between them that we're not using is not termed an intron, they're termed IVS. Because the exons are not being spliced together, they're being processed separately. Does that answer your question?
Yeah, kind of. I'm trying to understand. You said that the 18S and the 28S, they will be spliced separately, correct?
They're not being spliced together. They're processed separately. They're two different proteins. So normally when we go through transcription and translation, we go through transcription and we cut out all the introns and then we splice together the exons and then that RNA gets translated to become one protein. But since we're not making one protein, we're making two separate proteins for the two separate subunits of the ribosome. When we translate that, we're not, we're cutting out the introns, but we're not splicing the exons back together because they're making two separate things. So since they're not getting spliced together, those introns are termed IVS because they're not true introns.
Oh, that's the introns that turn into IVS, correct?
Yeah, because the exons are not being spliced back together.
Oh, okay. So on the example he gave us on figure 24, so the genes 18S and also 28S, in between there's like 5.8S, but I have two IVS. So each IVS belongs to each of the genes?
The IVS are the sequence that's in between the two genes.
Okay, I see.
Yeah.
Okay, thank you.
You're welcome. And he's probably not going to give you a picture. He's probably just going to ask you like, true or false, are IVS considered introns? And then you're going to say false, they're not.
So IVS are not introns, right?
Right.
Thank you.
You're welcome. Go for immuno. Yeah. Oh, really? Oh, really?
Yeah,
This is like what I would do. And then we would like take this and we would like put it on white.
Yeah, that's the idea. That is the idea.
I'm sorry, I have another question. For the ALU repeats.
For the what?
ALU repeats.
Yeah.
I'm trying to understand which one is the template strand versus the other coding strand, please.
So they can both be template and both be coding. So when he was asking that question, like which of the following strands is the template strand, he was talking about the RB1 exons, and you can tell which one is template and which one is coding in this particular like RB protein. Because if you look at the top strand, then you can see all of those exons there. And then if you look at the bottom strand, you don't see any exons. So that means that the bottom is the template strand and the top strand is the coding strand. But that doesn't mean that the bottom is only template. You can have the bottom be coding for separate genes. Does that make sense?
Yes, I just want to repeat that one more time again for me, please. Thank you.
What was that?
Can you repeat it for me again? The RB1 exons, the top versus the bottom strand, please.
So when he was asking you which of the following is the template and which of the following is the coding, he was only talking about that RB1, because the RB1 is showing you exons on the top strand and there are no exons depicted on the bottom strand. So since there are no exons depicted on the bottom strand, we can infer that the bottom strand is the coding strand for this RB1 protein, whereas the top, wait, did I say coding? The bottom is template. I don't know if I just mixed up.
Yeah. Okay, the bottom is template. Okay.
Because we don't see any exons. And the top is the coding because we do see exons. And the exons are those like little marks, the 1, 2, 3, like all 27. Those are the exons. So the top is going to be our coding and the bottom is going to be our template in this scenario.
Because of the X.
Yeah,
right?
because of the exons. Yeah.
Okay, perfect. The exons give me my landmark to know the coding strands, regardless of where they are located, correct?
Yeah, so like you could flip that, and then if the exons are on the bottom, then we would say that the bottom is the coding strand and the top is the template, because each strand can be coding. It just depends on what genes you're looking at.
All right, got it. Thank you so much.
You're welcome.
Thank you.
Yeah. Yeah. Correct. Yeah, so like when a cell becomes a cancer cell, you lose your tumor suppressor genes and you gain oncogenes, right? So if that happens and a cell becomes like a cancerous cell, it could also be beneficial to lose these DNA repair mechanisms, because if the cell repairs that DNA, then okay, I'm no longer a cancer cell. Yes, it's a cancer cell, and then it silences those genes because it's just like, this is more fun for me. I want to be a cancer cell. Bye. Yeah, so like what researchers have done is they're like looking at, so there's never going to be like a one size fits all cure for cancer. There's like too much variation between all cancer cells. There's too much variation between like breast cancer, pancreatic cancer, colon cancer, whatever. And then inside that breast cancer tumor, there might be like multiple different cells, like different types of cancer cells within one tumor itself. So there's never going to be like a one size fits all. So what researchers are doing are like trying to figure out like, okay, this specific cancer, what can I do to manipulate this cancer into killing it? So instead of doing something like trying to like a radiation, well, radiation is a mutagen. Okay, so like a mutagen is something that's going to destroy that DNA. So if the cancer cell still has DNA repair mechanisms intact, that might not work, because yeah, I can mess up its DNA, but then it could just fix it. So they have to like, if your cancer cells don't have these DNA repair mechanisms, then it's like, okay, I could use that and maybe it'll kill the cancer. And they found out that it did. So it's just like different therapies for different types of cancer cells. So like not all cancer cells is this going to necessarily work. But if they found out that that specific cancer cell type mutated or silenced its DNA repair mechanisms, then it's like, okay, let's try this therapy because it's probably going to work. They can't repair their DNA and they can't carry out vital functions. So then those cells are going to die. That's the example he gave you in class, but I'm sure there are other tumors as well. I'm just not a cancer biologist, so I'm not sure.
Hi, can I ask you another question? I'm sorry.
Yeah, no worries.
Can you explain to me again the V600E gene, please?
Yeah, let me go to it.
Thank you.
So where was it? Right here. Okay. So if you look at the sequence for valine, the first one, that GUA, that's pretty similar to the first sequence for glutamine, which is GAA. So in our BRAF protein, the BRAF protein is like pro-proliferative, so it signals the cell to keep proliferating, to keep dividing. So in that protein, we can have a mutation from valine to glutamate. And if you remember, like your amino acids, glutamate is negatively charged. So when our BRAF protein wants to be turned on, it gets phosphorylated. So if our cell wants to proliferate, that BRAF protein will get phosphorylated. And phosphate groups also have a negative charge. So since if we have a mutation from valine to glutamate, and glutamate is a negative charge, and a negative charge on glutamate looks like a negative charge on phosphate groups, that BRAF protein is going to think it's phosphorylated, it's going to think it's turned on, and that cell is just going to keep proliferating. And it's like an uncontrolled proliferation, which can lead to cancer.
Okay, thank you.
You're welcome.
There's 9, but he does this bullshit of 3, 4, 5, 6, 7.
Oh.
Like each top.
Yeah. Oh yeah.
I hate when
he
he
did it. Where it was, yeah. So.
Yeah, of course. Where did I saw that answer, and that's why I'm telling you cancer, but hold on. Yeah. I saw that somewhere, which is why I'm saying the answer is cancer, but I don't remember exactly where I saw that. The answer is cancer, and I will look for where I found it, and I will like post it or talk about it in my review, or you could email me so I can have your email and I'll send it to you. But the answer is cancer. Yeah. You're welcome.
Hi, I'm sorry.
No worries.
Do you mind go over again the DNA topoisomerase 1? There's an abstract that he said that we need to know about that.
Topoisomerase?
Yes, please.
Hold on, I'm finding it. Oh, here, okay. So what he wants you to know from topoisomerase is that when we use helicase to like unzip that DNA to access each single strand to replicate, we're creating torsional stress like more down from that like point. So since we're creating torsional stress like more down on the DNA, we need topoisomerase. So topoisomerase like skips ahead to where we're getting that torsional stress, and it's going to like cut a phosphodiester bond on one side. So when it cuts that phosphodiester bond, the DNA will spin around itself and then it will stop and reach the conformation that it wants to be in, which is 10 base pairs per turn. So once it goes back to every turn, so one turn every 10 base pairs, then we will reform that phosphodiester bond and then just keep repeating that process until DNA replication is over and there's no more torsional stress on the DNA. Because if we get torsional stress on the DNA, then that DNA can just break. And then for different types of cancer, we can use drugs to inhibit topoisomerase. So then that torsional stress will just build up and the DNA will break and then the cell will have to die. Does that answer your question?
Kind of, yes, a part of it, but there's a drug here, it's amrubicin.
Yeah. Yeah, that's what I was just talking about. So if we, for cancer cells, if we inhibit topoisomerase, then the torsional stress will not be relieved and that DNA will be damaged and can't be used any longer, and then the cell will have to die. So that's why we can use that as like a chemotherapeutic agent.
Okay, because it breaks it cuts the phosphodiester bond. Is that why?
So topoisomerase. So when we have helicase unwinding that DNA, it's creating torsional stress like more down from that spot, like more downstream on that DNA. So like if I'm trying to like unwind a curly like phone cord and I'm trying to straighten it, those like bunches are gonna get like super knotted at the end of that phone cord. So to prevent that from happening to the DNA, we have topoisomerase coming in and cutting one side of the phosphodiester bond so it can like spin around itself and reach back to its desired conformation, which is one turn every 10 base pairs. So once it gets back to one turn every 10 base pairs, and it's not like all bunched up, coiled up, then topoisomerase will reform that phosphodiester bond and keep on doing it until DNA replication is done so that there's no torsional stress destroying that DNA. Because if we build up too much torsional stress, that DNA will be damaged, no longer functional, the cell can't go on, the cell dies. So for that drug, it's a topoisomerase inhibitor, so we're not relieving the torsional stress and that cell will die, which is good for cancer.
Perfect. Thank you so much. Thank you.
You're
Thank
welcome.
you.
You're
Do you
welcome.
do you mind go over the mechanisms of RB functions again, please?
So E2F is a transactivator. So it recognizes a regulatory sequence in the DNA, and that DNA is for like histone genes or DNA polymerase genes. So it's like pro-proliferative, stuff that we need for our cell to proliferate. So E2F, since it's a transactivator, it's going to recruit a HAT. We know that HATs add acetyl groups onto the tails of the histones, which take away the positive charge. So if there's not a positive charge on the histones, they don't interact with the negatively charged DNA as much, which opens up our chromatin, which allows for transcription and translation of those genes to make proteins that we need for proliferation. So that's what E2F does, pro-proliferative. HAT, so that we can open up that chromatin and transcribe pro-proliferative genes. But we don't always want that to be working because we don't always want to be proliferating all the time. That's how we get cancer. So we need to have a mechanism that inhibits this action from happening all the time. So that mechanism is RB. So RB's function is to inhibit E2F until we want to proliferate. So RB recruits an HDAC. So the HDAC is making sure there are no acetyl groups on the histones. So the histones are still positively charged and they're interacting with the negatively charged DNA pretty tightly so that we have condensed chromatin, so we're not getting transcription of these pro-proliferative genes. So if we're not getting transcription of these pro-proliferative genes, we're not going to be dividing and making more cells. So RB is turned on when it's lightly phosphorylated. But if it's heavily phosphorylated, then RB is turned off. So when we do want to proliferate and we want RB to stop inhibiting E2F, we will heavily phosphorylate it. But if we don't want proliferation and we want RB to be inhibiting E2F, then it will only be lightly phosphorylated. And RB is defective in cancer. And they found it in the retinoblastoma cancer, so that's why they termed it RB.
Okay. In that case, in retinoblastoma, does that mean RB is heavily methylated?
RB is dysfunctional, so it doesn't even function to inhibit E2F.
Gotcha. Thank you.
All right. Okay, guys, if there's no other questions, everybody in the lecture hall left. So if you have a quick question, shoot. If not, we are going to head out and we are always available through email.
Appreciate all you do. Thank you so much.
You're welcome.
Bye-bye.